I'm glad to introduce our second speaker, Bradley Alicea, who is a senior contributor at the Open Science Foundation, OpenWorm Foundation, and is the head of the Orthogonal Research and Education Laboratory. His title is the Psychophysics of Non-neuronal Cognition. Welcome, Bradley.
Thank you. I'll put a related work in the chat in case you want to look into that further. Let me share my screen.
Thank you.
Okay, oops. So this is the Psychophysics of Non-neuronal Cognition, and thank you to the co-authors here who are here, but I'm presenting on their behalf. We're going to talk about an organism called Bacillaria. Actually, they're a class of diatom. And so the diatom is, I'll have to show these videos, maybe to give you a sense of what diatoms look like. They're single cell organisms that exist in multicellular colonies. So as you can see in this film, this is in the diatom, specifically Bacillaria in the wild, and they move in this way. They have these, we'll talk about how they move, and you can see that they're moving in water like this. And they fold and make all sorts of different conformational changes with respect to their shape. And then this is a single colony on the right, which is, and you're going to see in this example, one cycle of movement of a single colony. And so here you can see this single colony moving back and forth. This is something that's isolated in culture. And you can see how each cell moves against one another, and they make these movements. This is just one type of movement. As you could see from the other movie, they make all sorts of different movements and different orientations, but this is just sort of a controlled way of viewing this. So this is, we're going to appeal to something called psychophysics to talk about non-neuronal cognition. And I'll talk about what non-neuronal cognition is in a bit. It's a little bit of a controversial sort of hypothesis about behavior. But we're going to talk about possible psychophysical models. And so psychophysical models, we think of psychophysics as being a human thing. We think of performance, we think of all sorts of things like that. But then you can also observe psychophysics in different animals, non-human animal models. And so, for example, we can look at psychophysics of the mouse. We can look at their visual system. We can characterize performance quantitatively. We can also look at goldfish, and in goldfish, again, they have their visual system that we can attribute certain sort of quantitative laws to that. But can we also characterize diatom behavior and diatom movement in the same way? And that's kind of what I'm going to propose here. And so these citations at the top and the bottom are different psychophysics references, especially with respect to non-human animals. So these are diatoms, and probably most people in neuroscience aren't familiar with diatoms, but they're a very interesting group of organisms. They're an algae, so they exist in water, and they're single cell eukaryotes. So they're not prokaryotes, they're not archaea, bacteria, they're eukaryotes, just like us, but they have plant-like characteristics. So they have a chlorophyll element in the cell, and they have a silica cell wall. So their cell walls are rigid, but they also are eukaryotes. And so you can see on the upper right, you have these specific rapid diatom types. So the rapid diatoms, as opposed to some of the other types of diatoms you see over here, are these elongated shapes. They have here are the different attributes of the cell, these girdle bands, they have different valves, and this is basically the shape. And then on the bottom, you can see that they take on a number of different shapes. They almost like little machines or different plastic molds, but these are cells. And they come in rod shapes, flake shapes, and different 3D shapes. But we'll be talking about the rapid diatom today. It's interesting to note that the size is only about 10 to 80 microns. So that's very small for a single cell. And they exist in these colonies. Not all diatoms exist in the colonies, but these do, the ones we'll be talking about today. So we can analyze Bacillaria movement in culture, and it turns out you can culture diatoms, and this genus Bacillaria is one type of diatom that we can actually culture. So you can go out into the wild, into like a aquatic environment, and isolate these different colonies, put them in a culture. The culture conditions aren't too hard to replicate, and then you can observe these under a microscope. So here's a movie again of the single colony movement. And so again, the movement is isolated on a single focal plane, and you can see how they move back and forth. There's this oscillation in the colony. So each of the cells are moving. They're not moving independently. They're sliding against one another. And you can see that there's like a leading edge to this collective behavior, and then the other cells follow. So it follows a sort of accordion-like oscillation. There are also other phases of movement. So this is an idealized movement, but there are a lot of other phases of movement we can characterize. So there's a free sliding movement. There are changes in momentum during the oscillation. And so even in this small movement, you can characterize the different phases. But as you saw from the example of the wild colonies, that they move in different orientations in different ways that are very interesting. We can model this computationally as a series of coupled oscillators, of course, which I'll show you in this talk, but movement can vary due to stimuli. So we'll talk a little bit about the stimulus conditions and how that might affect the movement as well. So I'm going to talk now about non-neuronal cognition. And so it's a somewhat maybe controversial take on behavior and how behavior is executed. So there are a couple of citations that I've included that maybe are about in the last 10 years where they've tried to define non-neuronal cognition. And one definition from Leon in 2015 is that it's the total set of mechanisms and processes that underlie information acquisition, storage, processing, and utilization. So they think about it much like human cognition maybe, but they kind of give it this idea that you can observe this in systems that don't necessarily have neurons or sensory interfaces in the conventional sense. Baluska and Levin also published another paper where they contemplated what a non-neuronal cognition system might look like. And they make the point, one of the points they make is that the extracellular matrix is ideal as a memory medium. So they propose a bunch of different mechanisms for this. One of those is the extracellular matrix. They also suggest that there's a stigmergy that occurs where physical traces that are excreted in the extracellular matrix can be referenced and read into the future. And so one example from diatoms, Bacillaria in particular, are these mucilage trails. So remember I showed you these single cells sliding against one another. These single cells, they generate these mucilage trails between them. And so this is one of the things that helps them to stick together, but not, also be able to move sort of in concert. And so it's sort of like this viscous type medium in between the cells. And we can use these maybe as a memory matrix. These mucilage trails are secreted polymers. They completely encase the cell and are responsible for adhesion and interactions with the external environment. So let's think back to the sensory world inhabited by diatoms, and there is a great diversity of diatoms, but they're generally found in these different aquatic environments. So you can find them in marine estuaries, you can find them in shallow waters, you can find them, in places where they're sort of pelagic environments where you have a lot of biodiversity, and you just, go in and you can scoop up from the surface and you can isolate these diatoms. And so they're exhibiting characteristics of both an aquatic species, but they're also subject to light and other chemical stimuli. So one of those stimuli is CO2. And so we know from the literature that there's some effects of elevated CO2 on a natural diatom community. So we can observe that CO2 exists in the aquatic environment. These cells can sense CO2 presence of CO2 and concentration of CO2. And the reason it does so is because it affects the growth of the diatoms and their colonies. high levels of CO2, for example, reduces grazing pressure and has an effect on photosynthesis. So it's imperative for the cells to be able to sense CO2 in some way. And of course, they can sense changes in CO2 as well. They can also do things like sense motion, relative motion, and forces acting upon the colonies. So they exist in these water columns, and they're subject to water shear and other types of pressures like that. And so there are a couple of citations that I've included here on different things having to do with nutrients and environmental signals. And one of those signals is that they detect and respond to fluid motion, changes in fluid, the motion of fluid. So shear, osmotic stress, and iron abundance are all things that they sense and can respond to. Finally, they can also respond to light, as we mentioned previously. And this is a citation where they talk about light intensity in particular. And so CO2 and light availability affect diatom physiology. So CO2 and light in this case act in concert, although they don't have to. And so you can test, say in this paper at the top here, Hayden et al., they tested 3 light intensities. And so they found that low light levels affect physiology and distinct physiological traits. They can also localize sources of chemokinetic and chemotactic mobility. So they can sense chemical gradients, and they can use those as ways to find mates and other sorts of pheromone-guided searches. And so there's a fascinating physiology in the cells where they restore their cell size by reproducing. And so I'm not going to get into that today, but I will say that they use these sensory signals for that purpose. So we can go on to talk about a little bit higher level structure here. And one of the things we can talk about are these collective pattern generators. So these are the cells, again, they're moving against a substrate in this figure, but they're actually using these actin filaments and these myosin elements, and they're generating this adhesive mucilage. And they're moving against a substrate using these adhesives and this actin myosin complex. But imagine that this substrate is another cell, and you basically have a picture of how the Bacillaria colonies move. And so these actin and myosin systems interact between cell bodies, producing collective movements. So we will build our model of propulsion around this mode of movement, the CoPG. We'll propose a psychophysics model. We'll look at these oscillatory movements as a small subset of behavior, but allow us to view the CoPG circuit. And the psychophysics models that we'll propose will account, hopefully, for a wide range of behaviors outside of these oscillatory movements. So this is an example of a CPG. So the CoPG is based on a CPG, or a collective pattern generator. And so this is a circuit of a collective pattern generator in a neuronal system. So we have our neurons and we have our synapses here. So we have this network of neurons, and they're both inhibitory and excitatory synapses that play a role in setting up this collective pattern generator. And the collective pattern generator generates rhythmic motions such as insect movements of their limbs, also other types of pacemakers in the body. And so we can use that model. This is, of course, a neuronal model. But if we think about this now more broadly into the CoPG, which is a collective pattern generator, we can think of this in terms of different the cells being connected by sort of these points, these mucilage trails, and then they have a relationship between these different points that we can either arbitrarily measure or measure on the cell. And so we have these weights that vary by how the thing is moving, how the cells are moving against one another's strengths. There's like a weight of a certain strength between the 2 points. And so that gives us some way to model the movement of the colony. And so we can use a series of coupled sinusoids to model the ideal collective pattern generator. We can model like a pure sinusoid and then have overlapping sinusoids representing each cell, and then vary the distance between these overlapping sinusoids so we can get different degrees out of phase behavior. This simulates coordination or discoordination. We can also add noise to these sinusoids so we can get noisy sinusoids, which is helpful in some of the modeling the different phases of movement and maybe some of the noise that we observe. We can also look at the speed of movement or the derivatives of cell position. And so in the paper I put in the chat, we actually did some measurements of this movement in terms of its speed and its velocity and all of that. And so this can vary according to the effects of any given stimulus. And so one of the examples here are differences in stretch phase. So here's a colony and it's moving. You have this leading cell, this lagging cell, and then these intermediate cells. And you can see that when it stretches out, it doesn't just stretch out back and forth. These cells kind of bunch up about mid-movement. And so this can be modeled using this type of computational representation. We can also look at sensory thresholds. So in this case, each cell is receiving sensory inputs, which are these eyes, and differences in light intensity are measured by output movements in the weights. And so we can give it a certain amount of light input and it affects the movement of this network. We can also look at the signal to noise ratio. So this is useful in the dynamic sensing of water column physical properties. And that can also be measured by these output weights. So thank you for your attention. I just gave, I think, a short version of this idea. I'd be interested to see what you think. And if you like what you see, we can collaborate. Our group, DivaWorm, is a very highly collaborative group. We're a virtual group. We have a website and a series of GitHub repos. You can check those out. We have our weekly meetings on YouTube, so you can check those out. And I'd like to thank Thomas Harbeck for his observations of diatoms. He was the source of a lot of the videos shown in this talk. And check out his website as well. Thank you.
Thank you, Bradley. That was a really interesting and out-of-the-box talk. So that was fantastic.
Thank you.
So I have a couple questions from Najee. She's back online, but she's on audio with us. Najee, do you want to ask the question by yourself? Unmute and ask.
Sorry, she doesn't have a microphone either.
Okay, so I'll read the question. She asked, maybe because she missed the beginning a little bit, but she was asking, do you know the mechanism involved in to the sensitivity of the carbon dioxide? Are they sensitive to pH? Do they express channel involved in CO2 or O2 changes like the task channel?
I'm not really familiar with the biochemistry of it, but I mean, I know that they do have, I think it's probably somewhat similar to that, although I don't really know that much about it.
So I have a few questions, but I think I'll start perhaps with the title part, probably the more controversial part, right?
Yeah.
So which is are you equating cognition to just movement here, right?
Well, I mean, I guess the idea of cognition is information processing.
Mm-hmm.
So the idea is that you have movement, which is an output of what it's gaining. So it's getting sensory inputs, and there's some process within the cell, and
Mm-hmm.
it's producing an output.
Right.
And so the movements of course, can also be sort of, rhythmic, just kind of a set pattern, and then the inputs are changing that. So there's some information processing going on there. We don't know exactly what it doesn't necessarily matter what the mechanism is. It's just kind of a way to think about it. And so, that's the kind of thing, like it is kind of a metaphoric approach.
Right. Yeah.
But you can also use that paradigm to kind of build models around it and see the movement in a new light.
Right. I mean, I would say the same. We look at animal behavior, for instance, but we are using movement as a way to figure out if they're able to do something or not, right? So it is a element of cognition. That's why I was curious about your movement statement, because that's what mainly you were showing in your phenotypes there, which is really cool. I have one other question, but that's more related to the extracellular matrix. So maybe I'll come back to that in a bit. We have a question in the chat. What is the limit of non-neuronal cognition?
Well, the limit, you mean in terms of like the complexity of the behavior, or?
I assume so. Maybe we'll get a clarification in the chat, but I think that's a good start.
Yeah, I think it's probably, while it's a metaphor, you want to make sure your metaphor is somewhat rigorous. So you don't want to just call anything like cognition. There was a recent paper by, it was called Cognition All the Way Down, where they argued that cognition exists in cells and different subcellular mechanisms. I think, like, if you observe in a cell where you have these sort of intentional movements, I think you can interpret it as cognition. I'd be very careful about doing so. But I think the limit is basically where you have an input, some process internal to this thing, this cell or this organism, and then an output that's somewhat intentional. So, you want to distinguish between sort of stochastic movement, maybe like in maybe like Brownian motion, or something more intentional. And then that's where you kind of get into the definitions and kind of quantifying it.
Right.
So that's the challenge too.
Yeah. So maybe I'll piggyback on that with the extracellular matrix, because you're talking about how that's allowing them to tag with each other. And presumably that's dependent on the input and the output, right? So they're basing it on how they're interacting with each other, sensing their environment and putting those things down. Do we know much more about the intentionality there in terms of laying down that extracellular matrix?
I don't think it's very clear what the intention. I mean, they definitely generate this stuff, and then it's sort of sticks together. So there's probably a lot we don't know about, like what exactly is going on there. They do actually move, of course, intentionally, and the colonies actually fall apart after a while. So, we don't really know what's going on there. There could be just environmental factors at play where, the environment gets to a certain point where they just, or their age wise, they kind of fall apart. But there's definitely like a lot to learn there. There isn't a lot known about like some of the things I mentioned here, which are kind of speculative, but I think it's a really interesting system.
Right, yeah. Perhaps one last question before we have to wrap up. This is also for the question answer session. Can I also ask about the upper limit? As in, what's the most complex, sophisticated behavior an organism can do without neurons?
Well, I think it depends on like maybe, collective behavior is interesting because, like with bird flocks, for example, you have these and they have neurons, the birds have neurons, but the flocks don't have neurons. And so they have to coordinate their behavior in a way that goes beyond sort of the neuronal, standard neuronal suite. There's a lot of I mentioned stigmergy in the talk, and that's an idea from complexity science where they like a bunch of organisms, say like ants or something, will set up a bunch of things in the environment. So like these extracellular matrix trails, and they use them to read the movements of other organisms. And so ants can build trails through pheromones, laying them down and then having their neurons interpret those as like the way to go. And so you can do it that way too. And that makes for a lot more complex behavior than you would get from just a single brain.
Right, yeah. Fascinating material. Both of you, I've really enjoyed this session today. I'm glad in a little way that we were able to talk a little bit more because we missed out on our second speaker today. But this is fascinating stuff. We will post some of the links are on the chat, so feel free to follow up and perhaps communicate with Najee and Bradley through online mechanisms, because I think that's one of the cool things about being online, right? So they're able to do these and continue these discussions. So thank you again. This was absolutely wonderful and fun, and enjoy the rest of NMC. I'll see you soon.
Thank you,
William, for hosting us.
Thank you.